All right, we are going to get started with your last lecture for Module 1, and we're going to just continue the talk about membranes and fluid balance and things like that, so fluid chemistry. Now, when we talk about fluid chemistry, we're talking about an intracellular environment and an extracellular environment, and what I mean by that is that a cell has cytoplasm, and that's fluid inside the cell. And so that fluid has to compare with the environment that cell is in and has to compare with the fluid that makes that up. Excuse me. And so when we talk about the intracellular environment, we're talking about the cytoplasmic concentrations of fluid. And when we're talking about the extracellular environment, we're talking about the outside concentrations. So for example, if you think about a red blood cell in the bloodstream, okay, that red blood cell has cytoplasm. That's the intracellular environment. The bloodstream, which is mostly made up of water, and then plasma consists of a lot of different solutes, but that's the extracellular environment. So there has to be this balance in terms of solute and water concentration between the extracellular and intracellular environment. And things can move across the cell membrane, but in a selective manner. So a lot of membranes are very selective in terms of what they'll let cross. Some things can cross freely. So we need to talk about or review rather some passive and active transport mechanisms. So when we talk about passive transport, we are talking about things that don't use energy. So passive diffusion, facilitated diffusion, osmosis, filtration, those are all non-energy consuming types of transportation. The most common is going to be simple diffusion. So this is when solutes are moving from an area of high concentration to low concentration. And we call that the concentration gradient. So let's just say, for example, that over here, you have a very high concentration of sodium ions, and over here on this side of the cell membrane, you have a very low concentration of sodium ions. And if sodium is going to move from an area of high to low concentration, it's going to move from over here where it's high to over here where it's low. And molecules are in constant motion, so they may move back and forth before they reach that equilibrium. This is a good image here of what this looks like. So let's just say, for example, that this membrane is permeable to both water and a solute. That's not always the case. Usually smaller solutes are more permeable than larger solutes and other things like that. But in this case, for simplicity, this membrane is permeable to solutes and water. So we see a very high concentration of red ions on the side A and no red ions on side B. So it's very high on side A, very low, actually none on side B. So naturally, those red ions are going to move from this area of high concentration into this area of low concentration. So those are going to pass through the membrane until they are equivalent on both sides. And actually, while that's happening, you can see this blue ions, which are water molecules, also moving over to side A because they are very high on this side compared to this side. So they're actually going to move as well. Because that's water, we're going to talk about that in terms of osmosis in just a little bit, but it's the same idea. Okay, and then eventually over time, we reach the state of equilibrium. Physiologically, we see this a lot in our blood vessels in our cells. So we know that our blood vessels, particularly arteries, are always carrying oxygen to our tissues, right? And so our blood vessels, in terms of arteries, are very high in oxygen. So let's just say that this is an artery. Arteries are high in oxygen, and what they're high in, they are low in the opposite gas, which would be carbon dioxide. So this is high in oxygen. Our cells that make up our tissues are low in oxygen. They're waiting to be delivered the oxygen. And so the oxygen is going to move from this area of high concentration to this area of low concentration. Now look at the carbon dioxide levels. Because the cell is low in oxygen, it's going to be naturally high in carbon dioxide, which is a waste product of cellular respiration. And so that carbon dioxide is going to move from an area of high concentration to an area of low concentration. And that's going to be into the blood vessel. And what we will talk about when we get to the respiratory system and cardiovascular system is that then this carbon dioxide is going to be delivered to the lungs, and it's going to be exhaled. The next type of transport, passive transport that does not use energy, is called facilitated diffusion. It is very similar to regular diffusion, except this is meant for those particles that cannot cross the membrane on their own. And so in that sense, there might be carrier proteins or ion channels that those ions can move through. So for whatever reason, this gray and this pink, different types of ions cannot freely cross this phospholipid bilayer, and they need a specific channel or a protein to take them into the cell. And so, for example, this gray, let's just call it glucose, for example, because glucose moves into the cell via facilitated diffusion. Let's just say that this glucose is going to move into this carrier protein, and it's going to spit it out on the other side into the intracellular environment. And then same with the pink, except for they can freely pass through that channel. So these are just kind of helper proteins to help move things across that membrane. Still no energy being used here. When we talk about diffusion of water, we're talking about passive transport. So osmosis is the movement of water across a membrane, and we can safely assume that water can cross this membrane. And the interesting thing about osmosis is it's interested in water, but it's moving from an area of low solutes to high solutes. And I don't like to use the term high water to low water because it's really important that you think about it in terms of solute concentration. And the best analogy that I can come up with is that if you would like to dissolve salt in a glass of water, there's only so much salt that you can put into that glass of water to have all of that salt dissolve, right? So if you put too much salt into that glass of water, there might be some salt left over that never dissolves unless you add more water. So in cases like this, the water is going to follow the solutes. We need that level of water to be at a certain level to be able to dissolve all of those solutes that are available. What's going to happen here is that we have this high area of solutes here and a low area of solutes here. And let's just say that this purple is a protein molecule, and it's stated that that cannot cross the membrane. So we cannot do passive diffusion, right? Cannot just freely cross the membrane. So what needs to cross the membrane instead? Water. And so water is going to move from an area of that low solute concentration to an area of high solute concentration. And so that water is going to move from side B to side A. Notice how I don't like to talk about it's moving from an area of high water to low water because these water levels look essentially the same. So it's always good to think of it in terms of solutes. Another little rule of thumb to remember is that water always follows solutes. So wherever the higher solutes are, water is going to follow. Filtration is our next type of passive transport. So filtration is based upon something called hydrostatic pressure. And hydrostatic pressure is basically the pressure of water influenced by gravity. So gravity is a force here pushing down on this filter system, and that's going to force water through this filtration system. What it's also going to do is separate the solid material, which will get caught in the filter paper. And then the water will just be left, which is the solvent. So the solute will be left here, the solvent will be here. And it's driven by a gravitational force. Still no ATP, no outside energy. This is a natural gravitational force. And filtration will come in to play again a lot when we talk about the kidneys. So here's an example. So the gravitational force per se in the body physiologically would be blood pressure. So here's a blood vessel here. We have a pressure within this blood vessel, and we constantly have solutes moving in and out of that blood vessel. Now out here are just cellular tissues, just tissues, right? So maybe like fat tissue or something like that. Larger molecules cannot pass through those cellular spaces, but small molecules might be able to with some force. So that blood pressure might force these small molecules through that membrane, which will be the vessel wall, into that interstitial space or that intercellular space outside the body, now or outside the blood vessels. An example of this would be edema. So you've all seen somebody with very swollen like ankles or feet or something like that. And what happens with that is perhaps somebody with really high blood pressure has a great force coming through here with a higher rate of filtration. If they have high blood pressure, they're going to have more pressure filtering things out, right? And as more solutes come out, I said that that water follows solutes. So the more solutes that we have come out, the more water that's going to follow. And if this is just your general tissues, that's where fluid will accumulate. And that's what causes that edema or that swelling. Lastly, we have active transport, which is a ATP driven event. So different than the other four that we just talked about. There are different types of active transport, but we're just going to talk about active transport in its generic form for this class. But essentially, active transport allows particles to move against their concentration gradient. So before I talked about solutes moving from a high solute concentration to a low solute concentration. Well, what happens if we need to move it backwards? It's not with the concentration gradient anymore. It's actually against the gradient. So now we're going to move things from a low to a high concentration, and we're going to talk about why we need to do that. So it's very similar to facilitated diffusion, but we're moving from a low to high region using ATP this time. So this is a very good example. This is the sodium potassium pump. This is very important in muscle contraction and nerve stimulation. So naturally, on the outside of the cell, there's more sodium, and inside the cell, there's more potassium at resting. And naturally, because there's more sodium out here and there's more potassium in here, they're going to both want to move to an area of lower concentration. So they're going to want to move to a high to a low area. So sodium is going to move from this high concentration inside to a low, and potassium is going to move from a high to a low outside. That's natural tendencies. Now, when that happens, this causes an electrical difference, right? Or action potential threshold. All those words should sound hopefully a little bit familiar if you've already had 215, but that's going to cause an electrical impulse. And in terms of like muscle contraction, that will actually cause that contraction to occur. Now, these arrows are drawing the movement in the opposite direction because this is showing the active transport that resets the cell membrane. So again, naturally, things are going to want to move again with their gradient. Well, after that muscle contraction happens, in order to generate the next muscle contraction, we have to reset those ions to their respective sides of the cell that they started on. So imagine that this has moved with its concentration gradient. You would see more purple on this side and more pink on this side. And in order to have that be able to contract again, we need to reset that. So the purple sodium ions are going to move back out and the pink potassium ions are going to move back in, and then it will start over. But notice that's moving it against its gradient. It just got balanced, and now we're going to reverse that. But that requires energy. There's very important reasons why. So that sodium potassium pump is a really good example of the active transport. Now, when we talk about descriptive words to talk about these solutions with how many solutes they have, we talk about tonicity. And tonicity is basically just what the composition of that solution is. And we talk about movement of water typically when we talk about tonicity because we are describing the solute concentration. And so water is going to follow solutes. All right, so if the concentration of water and solutes inside the cell is equal to that outside the cell, we call it isotonic. So if let's just say we have a red blood cell in a bloodstream, right? The water and solute concentration in this red blood cell is the same as the water and solute concentration in that blood plasma. So we have no movement of anything, no net movement rather, and everything's happy, it's isotonic. An example would be 0.9% of sodium chloride. This is like if you go to the hospital and you need IV fluids to rehydrate you, that is what they're giving you. They're giving you an isotonic solution not to disturb your blood cells. Now, if that's not balanced, if it's not isotonic, it could be hypertonic or hypotonic. Hypertonic means high solutes. So hyper is high solute concentration in the solution compared to the cell. Hypotonic is going to be low solutes compared to the cell. And remember that water will follow solutes. So keep that in mind. We're going to go through a couple examples. Okay, so let's imagine this is a cell in the body, and this cell has 20% solutes, which means it has 80% water. That always has to equal 100. And outside the cell, we have 75% solutes and 25% water. Okay, and if water follows solutes, which means water is going to go towards the area of more solutes, which way do you think that water is going to move? Hopefully you said outside the cell. So water is going to move from this area of lower solutes towards the higher solutes. Now, what happens when you do that is this cell is going to lose water, which means it's going to lose volume, which means that it's going to kind of shrivel up and be dehydrated. So I want you to hang on to that idea for a second. Let's go over to the next example. So this cell has 80% solutes, this and 20% water, and that environment has 25% solutes and 75% water. So what's going to happen here? Water follows solutes. So, excuse me, wherever the more solutes are, that's where water is going to go. So which way is water going to travel? Hopefully you said inside the cell. So water is going to go from this area of 25 solutes into that 80 solutes. Remember, the more solutes you have, the more water you need to be able to dissociate those solutes into their ions. So if we're going to describe the environments of each of these cells, this cell is in a hypertonic environment because there's a very high solute concentration in that environment. This cell is in a hypotonic solution because the number of solutes is much less over here. So if it's a hypertonic solution, if a cell is put in a hypertonic solution, water will leave the cell. If that cell is put into a hypotonic solution, water will enter the cell. And that means that that cell is gaining volume, which means that it's going to swell. And this actually does happen in physiology in the bloodstream. So let's take let's look at isotonic first. So let's look at the middle one here. So normally, this is what your red blood cell looks like. It's biconcave, kind of looks like a donut. When that blood cell is in an isotonic solution, so it's in its happy state, your body's balanced, the solutes concentrations are equal in the cytoplasm of the cell to the outside environment, which in this case would be like blood plasma. And there's no net movement. I mean, you're always going to have movement of things in and out, but it's going to happen at an equal rate that nothing's going to swell or shrink too much. Now, what happens if we put that cell into a hypotonic solution? And what does that mean? Let's go back. Hypotonic solution means that there's lower solutes in that solution than there is in the cell. So if there's low solutes out here, that means there's high solutes in here. And if there's high solutes in here compared to here, water is going to enter that cell, right? Water follows the higher solutes. And look, what's happening is that red blood cell is swelling. And this one is actually bursting. That is called hemolysis. And that is a medical problem that actually can happen. Okay, what if we were in a hypertonic solution? So let's just say that this is a very salty environment compared to that cell. Okay, higher salts out here than there is inside the cell. Water is going to leave that cell, and then that cell is going to shrivel and it's not going to be functional either anymore. So that was placed in a hypertonic solution. So kind of relating this to physiology, when we get to red blood cells, we can talk more about this if this is confusing.